Every single mushroom sitting on the shelf at your local grocery store is actually, in a very real evolutionary sense, a closer cousin to you than it is to the lettuce sitting right next to it.
Which, I mean, that completely upends how most of us view the produce aisle.
Right.
Yeah, you look at a mushroom, it's, growing out of the dirt, it doesn't move, so you just naturally assume it's a plant.
Exactly.
But molecularly, that fungus shares a common ancestor with animals. It's something called Opisthokonta.
Opisthokonta. Okay.
Yeah, the plant evolutionary branch actually split off billions of years before that.
Wow. Well, welcome to today's deep dive. We have a massive stack of medical terminology sources in front of us, and we are unpacking the microscopic culprits behind infectious disease.
The big ones.
Yeah, the big ones. Specifically, we are looking at 3 foundational roots in medical terminology. So that's bacterio for bacteria, viro for viruses, and myco for fungi.
And it's a fascinating topic because it really changes how you look at the world around you.
Totally. And our mission today is to show you why that casual word we all throw around, like when we just say germs, is completely useless.
It really is.
The boundaries between these 3 kingdoms are, well, they're chaotic. They constantly break the rules of biology. And understanding their actual mechanics changes everything about how we survive them.
Because lumping them all together as germs is actually one of the most dangerous and honestly most expensive misconceptions in modern medicine.
Yeah, I bet.
When you understand the sheer mechanical differences between, a bacterium, a virus, and a fungus, you realize why treating them requires completely different battle plans.
Okay, let's unpack this. We all know they're different, but let's talk about the mechanics of why that matters. I always think of grouping bacteria, viruses, and fungi under the word germ as being kind of like pointing at a shark, a nuclear submarine, and a scuba diver.
Oh, I like this.
Right, and just calling them all swimmers because they happen to be in the water together.
That's a great way to look at it. The environment is the same. So in this case, your body is the water.
Right.
But their internal machinery is fundamentally unrelated. Like, take bacteria. Bacteria are self-sufficient, single-celled organisms.
Okay.
They have their own metabolism, they generate their own energy, and they reproduce through something called binary fission. So they are the scuba diver, in your analogy, swimming around under their own power.
Got it. Then you have viro, the viruses, which are like the exact opposite.
Completely.
They are essentially biological freeloaders. They aren't even cells. A virus is basically just a string of genetic code wrapped in a protein coat.
Which means they have absolutely no machinery of their own. A virus has to invade a host cell and literally hijack its internal factory to make copies of itself.
So they're the shark. We know they're the submarine.
Actually, in your analogy, they might be more of a sea mine that waits to attach to the submarine, but
Yeah.
then you have myco, the fungi.
Right.
These are eukaryotic organisms, which means their cells have a defined nucleus, so they're much closer to our own human cells.
Which goes back to the mushroom thing.
Exactly. And they range from single-celled yeasts, like Candida, to massive multicellular molds. And fungi digest their food externally. They secrete enzymes into their environment and absorb the nutrients.
So if I take my submarine and shark analogy a step further, this completely explains the real-world problem with the word germ.
How so?
Well, taking an antibiotic for a viral cold is like using a torpedo designed specifically to seek out a submarine's mechanical propeller.
Right.
But firing it at a shark, the shark doesn't even have a propeller. You're just blowing up the water.
That perfectly illustrates the mechanism of antimicrobial failure. Antibiotics target structural vulnerabilities that only exist in bacteria. Some antibiotics are designed to just obliterate the bacterial cell wall. Well, viruses don't have cell walls. They have protein coats.
So the antibiotic just bounces off, basically.
Pretty much. And other antibiotics work by gumming up bacterial ribosomes. Those are the little factories bacteria use to manufacture proteins.
Okay.
The drug binds to the bacterial ribosome and shuts it down, but it leaves human ribosomes completely alone because ours are structurally different.
And since a virus doesn't have its own ribosomes at all,
Yeah.
I mean, it's just using yours. Right?
Exactly.
Throwing an antibiotic at a virus does absolutely nothing to the illness, but it does wipe out the helpful bacteria in your gut.
Yes.
And it contributes to the whole global crisis of antibiotic resistance.
Because you just trained the surviving bacteria in your body on how to evade that weapon in the future.
Wow. Okay, so if these 3 things are so fundamentally mechanically different,
Yeah.
it kind of makes you wonder how science ever figured out how to categorize them in the first place.
It was not easy.
No. Looking through our sources, the history of taxonomy is basically just a series of hilarious identity crises.
What's fascinating here is that we were categorizing things purely by what they looked like through very, very primitive lenses.
Right.
Going back to your point about fungi being closer to animals than plants, Carl Linnaeus, the guy who's basically the father of modern taxonomy in the 1700s, he firmly stuck fungi right into the plant kingdom.
Naturally. I mean, it looks like a plant.
Right, and they stayed there in biology textbooks well into the 20th century.
Until, the 1969 Whitaker Five-Kingdom system that finally gave fungi their own independent status?
Yes, but the real aha moment didn't even happen until the 1990s.
That recent?
Yeah, with the rise of molecular phylogenetics. When scientists finally started reading the actual genetic barcodes of these organisms rather than just looking at their shapes, the entire map of life had to be redrawn.
And the exact same crisis happened with the bacteria root, didn't it?
It did. For most of the 20th century, the word bacteria was just the ultimate biological junk drawer.
The junk drawer, I love that.
It really was. If a microbe was single-celled, lacked a nucleus, and looked like a tiny blob or a rod under a microscope, scientists just tossed it into the bacteria bin.
Right. Until Carl Woese and George Fox came along in 1977.
Yes. They started comparing ribosomal RNA sequences across different microbes, and they were looking at organisms that lived in extreme environments.
Like really extreme. The sources mention boiling hydrothermal vents and hypersaline lakes.
Exactly. And under a microscope, these extreme microbes looked exactly like ordinary bacteria.
Yeah.
But when Woese and Fox actually looked at the RNA, the genetic sequences were completely alien. These organisms were as genetically distinct from standard bacteria as bacteria are from human beings.
So they just had to rip them out of the junk drawer and create an entirely new domain of life.
Yes, archaea.
Archaea. So it means the medical term bacteria today is actually a vastly more precise, narrow category than it was even 50 years ago.
Right. So fungi and bacteria were merely sitting in the wrong categories. The viruses, the viro root, they present a much deeper, almost existential problem for biologists.
Because they challenge the very definition of what it means to be alive. Because, if we go by the classic textbook definition of a living organism, you need a metabolism, you need to generate your own energy, and you need to be able to reproduce.
And as we established, a virus flunks that test immediately.
Right. So how can a virologist consider something a biological threat if it's essentially just an inert chemical package?
It sparks endless debates. If you go to virology conferences, there's always an argument.
I can imagine.
One camp calls viruses obligate intracellular parasites. They deliberately deny them the status of a living organism.
So to them, a virus sitting on a doorknob is, no more alive than a grain of salt?
Pretty much. It's complex chemistry, nothing more.
But the other camp looks at the execution phase. It's like a line of code on a flash drive isn't doing anything while sitting on your desk.
Good analogy.
But the second you plug it into a computer and execute it, it can rewrite your entire operating system. Once that viral genome unpacks inside a host cell, it directs protein synthesis. It replicates, it manipulates the cellular environment, it behaves dynamically.
Right. And just when the scientific community felt, sort of comfortable with that flash drive analogy, along came the giant viruses to completely ruin the paradigm.
Oh, the Mimi virus. I couldn't believe this story when I was reading the sources.
That's wild.
So it's 1992. Researchers in England were investigating a pneumonia outbreak, right? And they found this microbe inside an amoeba. And it was so massive that it actually absorbed a gram stain.
Which is crazy. Let's clarify what that stain means for the listeners. A gram stain is a classic lab test where a specific purple dye is applied to a sample.
Okay.
If a microbe absorbs and holds onto that purple dye, it's called gram-positive, and that usually indicates the presence of a thick, complex cellular armor made of peptidoglycan.
But a virus shouldn't have any cellular armor.
No, it shouldn't hold that dye at all.
So these researchers looked at this giant purple stained blob and just assumed, obviously, this is a gram-positive bacterium. And it literally sat in a laboratory freezer, completely misidentified, for over 10 years. How does the entire scientific establishment mistake a virus for a bacterium for a whole decade?
Because the fundamental unbreakable rule of virology at the time was that viruses are submicroscopic.
Oh, they had to be tiny.
Right. They had to be infinitesimally smaller than the cells they infect. So when French researchers finally sequenced its genetic material in 2003, they realized it wasn't a bacterium at all.
It was a virus.
Yeah, and it was roughly 750 nanometers across, which is an absolute behemoth for a virus.
And it wasn't just its physical size that broke the rules, right? When they read the genetic code, this mimivirus was carrying genes for protein synthesis.
Yes.
Genes that scientists previously thought were exclusively unique to cellular life. I mean, it totally blurred the line between inert chemical package and living cell.
Which really illustrates how incredibly difficult it was for early medicine to fight these threats. We couldn't clearly define them, and for most of human history, we couldn't even see them.
We were just flying blind.
Completely blind. You can actually track this blind struggle through the etymology of the medical terms themselves. Like the root viro, it comes from the Latin word virus.
Right.
But originally, that had absolutely nothing to do with microbes. It meant a noxious fluid, or a poison, or a venom.
Yeah, in the 1st century, the Roman Encyclopedia Celsus used the word to describe snake venom. It just meant a bad liquid.
Exactly.
But that raises a major question for me. Let's fast forward to Louis Pasteur in the 1880s, working on rabies.
Okay.
He's using the word virus to describe the agent causing rabies, but his microscope wasn't powerful enough to actually see a viral particle.
True.
So if he couldn't see it, how did he even know there was an infectious agent there at all?
Yeah.
Why didn't he just assume the brain was malfunctioning on its own?
Well, he did it through rigorous serial transmission. Pasteur couldn't see the rabies agent, but he could take tissue from an infected animal, inject it into a healthy one, and reliably reproduce the disease. So he proved there was a replicable, transmissible agent at work, even if it was completely invisible to his instruments.
But he still thought of it as essentially just a tiny bacterium that he couldn't resolve in his lenses, right?
Yeah, he didn't realize it was a totally different category. The actual conceptual leap realizing viruses were physically different from bacteria came from that brilliant filter experiment with tobacco plants.
Chamberlain filter.
Yes. In 1892, Dmitry Ivanovsky was studying tobacco mosaic disease. He took sap from a diseased plant and pushed it through this unglazed porcelain filter.
And the pores in this porcelain were so microscopic that they were guaranteed to trap every single known bacterium.
Exactly.
But when he took the liquid that dripped out the other side and rubbed it on healthy plants, they still got sick.
Right. But Ivanovsky couldn't let go of the bacterial paradigm.
Really?
Yeah. He concluded that his filter must be broken, or that a bacterial toxin was just slipping through.
He couldn't see past his own assumptions.
Exactly. It required Martinus Beijerinck, a Dutch microbiologist who repeated the experiment in 1898, to look at the data and say, no, the filter works perfectly. We are dealing with a fundamentally different, completely invisible kind of infectious agent.
And Beijerinck called a contagious living fluid.
Which marks the true birth of modern virology.
That is so cool. Meanwhile, the root bacterio comes from the Greek word bacterian, which means a little rod or staff.
Purely descriptive.
Right, based on what those early microscopes revealed.
Yeah.
But there was this massive lag between spotting these shapes and understanding their molecular mechanics.
Oh, huge lag.
I mean, look at the 1918 influenza pandemic.
Right.
50 million people died worldwide. And doctors were deploying masks, quarantines, and all these epidemiological tools to fight a virus 15 years before human eyes ever actually isolated the influenza virus.
Which didn't happen until 1933.
Yeah, using ferrets. It's the ultimate example of medical practice being forced to outrun molecular science.
If we connect this to the bigger picture, the struggle to lock down the rules of disease was intense. Like in the 1880s, Robert Koch established Koch's postulates.
Right.
And those became the absolute gold standard for proving a specific microbe caused a specific disease. The rules were very strict. You had to isolate the microbe from a sick patient, grow it in a pure culture in a lab dish, use that culture to infect a healthy host, and then re-isolate it.
But those rules completely fail for viruses.
They do.
Because you can't just swab a virus onto a petri dish of agar jelly and expect it to grow.
Yeah.
Without living host cells to hijack, a virus does nothing.
And honestly, the rules broke down for certain bacteria, too.
Really? Like what?
Well, Mycobacterium leprae, the bacterium that causes leprosy, still refuses to be reliably cultured in a standard lab dish, even today.
Wow, even today?
Yeah. And Treponema pallidum, the bacterium responsible for syphilis, that wasn't continuously cultured in a lab until 2018.
2018, that's just a few years ago.
They require incredibly specific, complex biological environments to survive. So the fundamental rule book for defining disease causation has been full of exceptions for well over a century.
It just shows how these microbes wage war in ways that are wildly unpredictable. And they don't just attack humans, they attack each other.
Tackle it constantly.
And understanding that microbial warfare gave us the greatest medical breakthrough of the 20th century.
You're talking about Alexander Fleming's discovery of penicillin in 1928.
Exactly, the classic story. He comes back from vacation, looks at his neglected petri dishes, and finds a stray mold has contaminated a culture of staphylococcus.
Right.
The mold was a microorganism, a fungus called Penicillium notatum. And the bacteria was obviously a bacterial organism.
And the fungus was literally slaughtering the bacteria in a ring around itself.
It was biochemical warfare playing out on a glass plate.
It was. The fungus was secreting a chemical weapon to defend its territory from the bacteria. Fleming realized what was happening, and eventually humans learned to extract that fungal weapon and mass produce it.
So the entire antibiotic era, every time we talk about fighting infections or worrying about superbugs, it all traces its lineage back to one fungus defending its turf against a bacterium.
Here's where it gets really interesting, though. The interactions go so much deeper than just warfare.
Oh, definitely. In some cases, these microbes are the literal reason we possess complex life at all.
Exactly.
Lynn Margulis formalized the endosymbiotic theory in 1967, and she proposed that roughly 1.5 to 2 billion years ago, an ancestral eukaryotic cell engulfed a free-living bacterium.
But instead of digesting it for food,
they formed a partnership.
Yes.
The host provided protection, and the bacterium generated energy. And over billions of years, those engulfed bacteria evolved into the mitochondria in our cells today.
And the mechanism that proved Margulis right, the undeniable aha moment for biology, is found in your DNA.
Tell us about that.
Well, your cellular DNA is housed in the nucleus. But if you look inside your mitochondria, they have their own completely separate circular DNA.
Circular DNA being the exact signature of a bacterium.
Exactly. They retain their own genetic barcode separate from the human genome.
That is wild. You're quite literally walking around powered by billions of domesticated bacterial descendants right now.
Which means bacteria isn't just an external pathogen, it is the engine of your own biology.
Wow. Okay, so because these microbes are so deeply interwoven with our biology and wage war so differently, the language we use in the emergency room had to evolve to reflect that precision, right?
Right. The terminology isn't just academic, it dictates the immediate battle strategy.
So you see these roots combined with specific suffixes. Like the suffix allosis means an abnormal condition, giving us mycosis for a fungal infection.
Yes. And the suffix amia refers to a blood condition. So if an organism breaches the bloodstream, a doctor diagnoses bacteremia, viremia, or fungemia.
And if that breach triggers a massive systemic immune response, the root septo combines with aemia to give us septicemia, right?
Exactly, which is a life-threatening state of blood poisoning. You also see roots like proto giving us protozoa, which are single-celled eukaryotes like the parasite that causes malaria.
Right, which is a stark reminder that beyond bacteria, viruses, and fungi, there's a whole fourth microbial kingdom of threats.
There is. So what does this all mean for us today? Why is this extreme precision knowing instantly if an infection is bacterio, viro, or myco? Why is that the defining public health issue of our decade?
That's a great question. And from the sources, it all comes back to antimicrobial resistance.
Yes, it does.
These organisms are adapting to our weapons, and they are doing it along these exact root lines. Viruses rapidly mutate to escape vaccines and antivirals. Bacteria acquire genetic mutations to withstand antibiotics, creating untreatable superbugs.
But the fungi are adapting in a way that might be the most alarming development of all.
The sources highlight Candida auris, right?
Yes, Candida auris. It's a multi-drug-resistant fungal pathogen that practically didn't exist in the medical literature before 2009.
Really? Just out of nowhere.
Pretty much. And now it's causing lethal outbreaks in hospitals worldwide. And mycologists like Arturo Quesadoval have put forward a deeply concerning hypothesis about why this is happening now.
It has to do with temperature, right?
Yes. Historically, mammalian body heat has been our greatest invisible shield against fungal infections. Most fungi thrive in cooler environmental temperatures.
So human body temperature is simply too hot for them to survive and replicate inside us?
Exactly. But the global climate is warming.
Oh.
Yeah. As the environmental temperature rises, fungi are being forced to adapt to hotter conditions just to survive in the wild.
So Quesadoval suggests that as they acclimate to a warmer planet, they are inadvertently breaching the thermal barrier that used to protect mammals.
Right. If fungi adapt to thrive at 98.6 degrees Fahrenheit, we are going to face entirely new classes of systemic infectious diseases that our bodies have absolutely no natural defense against.
That is terrifying.
It really is.
This means when a patient develops septicemia in the ER, the diagnostic muddy waters have to be cleared instantly. The doctor needs to know exactly which kingdom they are fighting.
Because reaching for an antibiotic torpedo when you need an antifungal medicine wastes precious time.
The root word dictates the drug, often within the hour, because the biological armor of each kingdom demands a totally unique weapon.
Exactly. We aren't fighting a monolith called germs. We are navigating 3 entirely distinct biological worlds, each with its own evolutionary history and its own rules of engagement.
Absolutely. Well, before we wrap up this deep dive, I want to leave you with one final, bizarre historical mystery from our sources.
Oh, this is a good one.
We've talked about how fungi might be adapting to threaten our future, but they may have played a highly deceptive role in our past.
The Salem witch trials of 1692.
Yes. In 1976, a psychologist named Linda Caparel proposed a fascinating theory. She suggested that the Salem witch trials might actually have been caused by a myco outbreak.
Specifically, ergotism.
Right. Ergotism is poisoning caused by a fungus called Claviceps purpurea, which infects rye grain.
And the mechanism here is chemical. When humans consume rye contaminated with this fungus, it produces potent alkaloids that are chemically similar to LSD.
It causes brutal convulsions, muscle spasms, crawling sensations on the skin, and intense, terrifying hallucinations.
So, to a 17th century Puritan village, an outbreak of ergotism would look absolutely indistinguishable from demonic possession.
It makes so much sense when you hear it.
It's an incredibly vivid, compelling narrative. However, historians and toxicologists heavily dispute it.
Yeah, they push back pretty hard.
They point out that the specific symptoms recorded in the court documents and the precise timing of the events don't actually align with how ergot poisoning behaves mechanically in a population.
So it remains more of an intriguing historical myth than settled science.
Right. But it's such a perfect caution against our modern desire to retroactively diagnose history with microbiology.
Totally. We want everything to be as clean and binary as a broken bone on an X-ray. We want to look back at the chaos of Salem, or even just the sniffles we have today, and just point to a single jagged line and say, there's the germ.
But the microscopic world doesn't play by our rules.
No, it doesn't. It's vastly more complicated, profoundly messy, and constantly evolving around us. Keep questioning the invisible forces shaping your world, because the closer you look at the roots of life, the more fascinating the picture gets.
Well said.
Thanks for joining us on this deep dive. We'll see you next time.